Thank you all for being here today. I really appreciate you showing up. And it's a huge pleasure and honor to welcome Dr. Ben Langmead, professor of computer science at Johns Hopkins. Just awesome to have him here and to get his talk, which is going to be on pan-genomic advances for fighting reference bias. I'll give a brief intro and then get out of the way because you're all here to hear him and not me. So I'm just going to read a few things just so I don't forget anything I wanted to mention. But basically, for those that don't know Dr. Langmead, he builds widely used methods in software analyzing high throughput genomics data. He earned his PhD at the University of Maryland in computer science in 2012. And I would say Bowtie and Bowtie 2 are two of his more widely used computational tools, but he's been involved with several others. Near and dear to my heart, things like metagenomic analysis, Kraken 2, et cetera. And a couple things I'll just read off here. The Bowtie paper won Genome Biology Outstanding Paper Award in 2009, which is well deserved. His work has been highly cited, I think over 100,000 times. And he's got some honors here I just want to mention. He's got the Sloan Fellowship and NSF Career Award both in 2014. I got a Johns Hopkins Career Impact Award in 2025. And just as a final note, again, I don't want to belabor this. I could go on for a lot longer for sure. I would just say that Ben has a ton of teaching materials online, incredible amount of teaching materials, has a huge commitment to teaching and materials related to kind of string algorithms. Just check it out if you haven't. It both on YouTube, but also slides and Jupyter notebooks and all of that. Just a massive amount of effort that I've grown to appreciate. And in fact, these materials helped inspire a class that I'm teaching now at Rice called Genome Scale Algorithms. We kind of saw that and kind of drew inspiration. So thank you, Ben, for that material. Awesome. But I'll get out of the way now. And again, let's welcome our speaker to talk about pan-genomic advances for fighting reference bias. Thank you, Ben.
Thank you very much, Todd. It's great. So thank you very much for having me. This is my first trip to Rice and Baylor, and I'm excited to talk to you about this topic. I know there are trainees in the audience, and I think one of the things I want to emphasize to the trainees today is that what I'm talking about today is something that was not part of my training when I first started, simply because we didn't quite have the tools yet to fully address this reference bias problem. But let me dive in and tell you more about what I mean about this problem and what those tools are for fighting them. And it will even give me a chance to talk through some of those teaching materials that Todd talked about, though I promise not to bore you. Okay, so early in our training, we probably learned how to use reference genomes, right? And you've probably had to download a reference genome and called it by one of its many names, like HG19 or GRCH38 or CHM13T2T, blah, 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 all these different names that we have for reference genomes. We also know that if we were to crack open these files and look at what the reference genomes actually are, it turns out that they are what we as computer scientists would call strings. They're strings of letters, A's, C's, G's, and T's. And in one somewhat comical illustration of just how long the human genome string is, there was an exhibit in London. I'm told it's not there anymore, so don't go looking for it, but where they had a printed out version of the human reference genome in a bunch of bound volumes. So that's what you're seeing on the right-hand side there. Okay. Since then, right, since the days of when we first got our reference genomes, there's been a huge number of sequencing-based studies that have uncovered more and more genetic variation. I'm going to continue to use human as an example, but we can think of other species as well. But for human, at least, we've had short-read sequencing efforts like the Thousand Genomes Project. We've had long-read efforts like the Human Pangenome Reference Consortium, the T2T sequencing project, HGSVC, there's others I could list. And the Human Pangenome Reference Consortium, for example, if we were to look at what they can offer us today, they can offer us something like 466 high-quality human reference genome haplotypes, right? Not quite as high quality as T2T, but close, a lot closer than we used to be able to get. So knowing now that we have all of these resources available, you might think to yourself that if we are going to analyze one new sequencing data set, maybe it's data from a patient with a disease, it would be a shame not to use all this information that we have about genetic information. So maybe we should. On the other hand, I can even say this a different way and say, if we don't use more of this information when we go to analyze data from a new person, we know we'll make mistakes. We know there's certain kinds of mistakes that we will make if we just use a single linear reference genome. And generally speaking, we try to put the label that we put on all these different kinds of mistakes that we might make, we tend to call them reference bias. The reference is just one single linear reference. It's not identical to the genome of the person we're studying, of the sample we're studying. That leads to mistakes. So let me show you some pictures of what these mistakes might look like. So when we go to place reads with respect to a reference genome, let's say using a read aligner, then some of the reads will cover places in the donor's genome that have non-reference alleles. Okay, so if the reference has all reference alleles, some of the reads might have the reference allele, or depending on what genetic variants this person has, some of them might not. And so if the read contains these non-reference alleles, the read aligner, which is trying very hard to solve exact matching problems and other pattern matching problems, is sometimes just going to be fooled. It's sometimes just going to be defeated. And it's not going to be able to find the true point of origin for that read just because it's burdened with all these non-reference alleles. The first place this was specifically described that I can recall is in early days of next generation sequencing when people were doing allele-specific expression. So they were trying to study whether RNA sequencing reads, short RNA sequencing reads, tended to come from one of the haplotypes or the other, the maternal or the paternal copy of a gene. And sometimes what they would find is that either the maternal or the paternal copy might have more non-reference alleles than the other. And in which case the read aligner is slightly biased away from finding that evidence that maps to the non-reference alleles. Okay, in this example that I'm showing on the left here, maybe it's not such a hard one because there's only one non-reference allele that I've showed on the maternal copy. But what if the situation is more dire, like maybe this example on the right now, there's a densely packed set of three non-reference alleles on the paternal copy. Maybe now the read aligner is just unable to find a seed for its seed and extend algorithm, and it just fails to find alignments for those reads. Okay, in this case, that's perfectly confounded with what you're trying to study, right? Because what you're trying to study is allele-specific expression. You want to know if more reads are coming from one copy or the other. In this case, that's confounded with the fact that the different copies have different burdens of non-reference alleles. So that was the first sort of instance. This is the citation at the bottom that I can remember reading the term reference bias, meaning what it means with the meaning that I'm using for this talk. And so that paper is a good 16 years old now. So we've had this idea for a long time. That's not the only form reference bias can come in. So for example, some regions, important regions of the genomes we want to study are hypervariable. So for example, the MHC region in the human reference genome. I'm showing you here what a short read pileup might look like in the MHC region of the human genome. Maybe not everybody here looks at IGV plots, but I'll just tell you this plot looks bad. This is not a good looking IGV plot because the reference genome is simply not similar enough to the person who's being studied in this case. And so a lot of the reads fail to align and you get troughs. You get these sort of peaky trophy, and these gaps are bad news. Okay, here's another example. I like to show this one. It's a bisulfite sequencing assay. So this is bisulfite sequencing of different mouse strains, and there are reference genomes available for both of the two mouse strains that are being compared in bred strains. And depending on which of the two you use as your reference genome, you either see a differentially methylated region in one direction or a differentially methylated region in the other direction. And the truth is, there's no significant differential methylation in this region, right? So both of these are spurious, and the direction of the spurious signal depends on which one you pick as your reference genome. Totally a nightmare, right? This is very bad news from the perspective of trying to do good and effective bioinformatics. I can show examples that use long reads as well. The phenomenon is quite similar, except it tends to be more significant in places where there's structural variation and copy number variation. Here's an example of a paper. I get to work with awesome people like Fritz, who's on this paper here. And so we see this for long reads as well, though in different ways compared to short reads. Okay, so having said all of this, we want to not accept this. Like we want, in this generation of trainees, we want to be trained in such a way that we can avoid a world where our sequencing-based diagnostics and therapeutics are differentially effective depending on whether the donor genome is a good match to the reference genome or not, right? So regardless of the person's genetics, and regardless of whether we're looking at a hypervariable region versus a not so hypervariable region. Okay. And if I can slide over slightly to the world of metagenomics as opposed to the world of analyzing one genome at a time, we also want to have similar benefits when we go to study genomes from across the tree of life, right? Like similarly, we want to be able to cover in our metagenomic databases many, many, many strains so that we're not going to fail to identify when a read seems to come from a particular species. And of course, our databases are now piling up with many, many, many wonderful assemblies of different species as well, not just human. So we want to live in a world where we don't worry too much. A short way to say it would be we want to live in a world where more is better, where adding more reference genomes to our compendia is actually giving us more accurate results when we analyze our sequencing needs. And so I'm here to tell you we can do this. And so there's basically two tools I would like to review. And these two tools together are going to enable, again, the next generation of bioinformaticians to do this regularly. Tool number one is pangenomes. I'm not so worried about giving you an exact definition of what a pangenome is here. You could imagine that when I say pangenome, I mean something as simple as a collection of linear genome sequences, not related to each other, not trying to relate them to each other in anything like a graph or a multiple alignment, just sort of letting them be sequences, leaving them alone. That's one definition. That's actually my favored definition, and I'll tell you a little bit more why. But another way would be to put them together in some way, perhaps multiply aligning them, and then letting the multiple alignment guide um, how you should collapse them into a graph shape, so a graph pangenome. These are both perfectly viable ways of building a pangenome and thinking of a pangenome. It could be just a collection of sequences. It could be something like the generating graph, such that all different ways of walking through the graph are the different sequences in the pangenome. The other tool I want to talk about, and this is where I'm going to have to go into teacher mode a little bit today, is the Burrows-Wheeler transform. The Burrows-Wheeler transform, which is a weird idea, but we're gonna tackle it. It's not going to be too hard. We're going to tackle it. This idea is sort of the wellspring of all of the best ideas we have for how to make use of these many sequences in the pangenome. Um, it's been with us for a long time. We've used it in tools like Bowtie and BWA for many, many years since the early days of second generation sequencing. What you might not know is that it's also the perfect tool for pangenomics, right? Almost unmodified. It's basically also the perfect tool for pangenomics. And the two directions in which the literature has taken the Burrows-Wheeler transform to adapt it to pangenomes are shown here. This direction, which is the one I'll emphasize today, but then another direction toward Wheeler graphs, which is more appropriate for graph-shaped pangenomes, which I'll just touch on today. But mostly I'll be talking about this left-hand fork in the road toward what's called the R index. Okay, so now we have to tackle the Burrows-Wheeler transform. So what is it? Why is it a useful tool for what we're trying to do here? Well, the Burrows-Wheeler transform is really just a way of taking the letters of a string. So let's take this thing on the left here and say that that's our input string. It could be a genome. It could be a collection of genomes. It could be many genomes, and we just concatenate them together, and that's the input. That's fine too. And we simply are taking the letters of that string and putting them in a new order. That's it. It's really just a permutation of the letters in a string. Okay, and in particular, we're putting them in order according to the alphabetical order of their contexts, the alphabetical order of what comes to their right in the original string. Why would we do this? Like, why would we even want to put something in a new order? It's in that order for a reason. That's the order that evolution gave us, right? Why are we trying to put things in a new order? Well, maybe a reason becomes more apparent if we think of an analogy. So, like, let's say I gave you a deck of cards, and maybe the deck of cards were in order, like this. And I said, let's play a game of poker. You'd probably be like, I think you should shuffle those first, right? Because otherwise, we're about to play a very boring game of poker where everyone has a straight flush, right? So, probably you'd want to take these and shuffle them. On the other hand, if they're shuffled, but I tell you there's only 51 cards here, one is missing, please tell me which one is missing. You'd probably want to put them in order, right? That would help you because once they're in order, it's very easy to see which card is missing. It would be kind of hard otherwise. And in general, in computer science, we're often concerned with taking data, putting it in a new order, because as soon as it's in that new order, suddenly new kinds of questions are easily answered. And same is true with the Burrows-Wheeler transform. We use the Burrows-Wheeler transform because when we put the string in this new order, suddenly questions about substrings of that string, whether those substrings are there and where they occur, are easier to answer. Another way it's often visualized is this way, by way of the Burrows-Wheeler matrix. You can think of this as this column here is the Burrows-Wheeler transform. These are the right contexts of all the things in the Burrows-Wheeler transform, and you can sort of put them in a nice alphabetically ordered matrix. I sometimes call this like the operational definition because you can actually build it this way if you wanted to. You can generate this matrix and sort it and then take the last column. Okay, so once we have the Burrows-Wheeler transform, we take that, we take a few extra data structures, and it's probably too detailed today for me to talk about those data structures, but let me take the first of a few opportunities I'll take today to say, please check out my YouTube channel because I've got a lot of teaching materials that cover all these details, far more details than you would ever necessarily need to know, but including what are these extra data structures. But let's just say we take the Burrows-Wheeler transform and we can use it as an index by driving a process that looks like this. So if we have a pattern and we have our Burrows-Wheeler matrix, this is the Burrows-Wheeler transform, it's the last column. We can, in a series of successive steps, take each letter of our pattern, starting from the right-hand side, and we can match longer and longer suffixes of our pattern. When I say match, I mean we're finding all the rows of this matrix that begin with that suffix. So here's the shortest suffix, which is just A. Here's the next shortest suffix, which is BA. Here's the next longer suffix, which is BA. Here's the longest suffix of them all, which is ABA. By the time we're finished, we can conclude immediately that there's two occurrences of ABA inside the string, which is true. That's one kind of, so each step of this process, I'll call that a match query. So we have to do that match query repeatedly for each letter of our pattern. And then once we have identified that there are those two matches, there's probably one more question we want to ask, which is, well, where were they? Where in the genome did those matches occur? That's a separate kind of query. We call that a locate query. So the locate query in this case would identify that they are at offsets. I'm going to be a computer scientist and use zero-based offsets, offsets 0 and 3 in the original string. Okay, so those are the two basic kinds of queries. If we can do those two queries, if we have an index and it can fit in memory and we can do those two queries and we can do them quickly, we're off to the races. We can do the sorts of things that read-aligners need to do with genomic data. Okay, but I'm telling you also that this is useful for pan-genomes. But I have to explain that a little bit. Like you might know that the Burrows-Wheeler transform is at the core of tools like Bowtie and BWA. So we know that that's at the core of tools that we already use to analyze individual, reads with respect to individual reference genomes. But why is it also useful for pan-genomes? So one thing I'll point out is that when the Burrows-Wheeler transform was initially invented, it was invented by two British computer scientists who were working at a company called Digital Equipment Corporation. They invented it for the purpose of compression, not indexing, but compression. Compression and indexing, it turns out, aren't that different. They're actually pretty close to the same problem, right? They're both about taking a big complicated data set, removing as much of the redundancy as you possibly can, but then leaving enough breadcrumbs so you can get back to the original if you need to. So if I were to show you now a real pangenome, like a really sophisticated pangenome. Okay, just kidding. That's not a real pangenome. It's just a toy example. But if I show you this toy example, I can demonstrate to you what it is that the Burrows-Wheeler transform is really doing in terms of compression. So here's just, imagine I've written it along 3 lines, but it's just one big long string I have here. So it's just row, row, row your boat repeated 3 times. Don't worry too much about this $ at the end. That's a technicality. And you'll see, obviously, this is repetitive, right? And it's repetitive in sort of 2 dimensions even, right? So vertically, it's the same thing repeated 3 times, right? So that's repetitive in that sense. But then even within one of these rows, it's repetitive because the word row is repeated. This is maybe a bit like human genomes, right? If I was indexing many human genomes, they would of course be similar to each other because unrelated humans have extremely similar genomes. But also, as we know, individual human genomes are internally repetitive. So similarly, my example has 2 different kinds of repetitiveness. So let me focus on just the W's for a moment. And I want to make the point that when we go to reorder things according to the Burrows-Wheeler transform, right, we're going to bring together the letters that have similar right context, right? The letters that have similar stuff to the right are going to come together in the Burrows-Wheeler transform. And the point I want to make is that the things that come together tend to be equal to each other. So for example, these W's, if you look at their right context, these W's, their right context is underscore R, and these W's, their right context is underscore R, and these W's, their right context is underscore Y, which is pretty close to R alphabetically. So all those right contexts are close to each other, which means that when we do the Bros-Wheeler transform, the W's should come together. And so if I just show you the full Bros-Wheeler transform of this entire string, it looks like this. And sure enough, there's our W's. They all came together because they all had similar right contexts. But it wasn't just the W's that came together, right? This happened all over the place, right? This thing here is this Burroughs-Wheeler transform string is what I like to call runny. It has lots of runs in it, and where run just means a lot of equal letters in a row. And in fact, if we just take it and we encode it according to the runs, using something called run length encoding, that just means like instead of writing these 9W's in a row, let's not waste our breath writing it 9 times. Let's just write W times 9 as a little placeholder for how many times W occurred. If we do that to this string, then we end up with these 15 things, right? 15 instances of a letter and the number of times the letter was repeated. And so roughly speaking, it's a bit like we took the input, which was 64 letters long, I counted it, and we turned it into 15 runs, which was kind of like compressing it. And if we just let the compression ratio be the number of letters in the original divided by the number of runs, we get a compression ratio of over 4, so 4.27. Okay, that's a good compression ratio. In fact, one thing that I'll point out about it is it's even better than you might expect if we had just taken advantage of the vertical kind of repetition, right? If we had compressed it just vertically, but not horizontally, we would expect our compression ratio to be basically 3, right? Because that's just 3 things repeated. This is better than 3. That's because we took advantage of both, right? We took advantage of the vertical kind of repetition and the horizontal kind of repetition in this compression procedure. Okay. This isn't really a pangenome, as I said before. So now let me show you a real pangenome. Okay, it's still not a real pangenome, but this one at least has mutations, all right? So, right, because if we're going to build a pangenome, we're not going to literally build it over identical sequences. That would be silly. There'd be no point. We want to build it over sequences that are similar but not identical. So here's an example where I have 3 sequences that are similar but not identical, similar to the first example. If I take the Bros-Wheeler transform and then I run length encode it, I get a result that is still pretty good. Not as good as the previous slide, which stands to reason because it's simply not quite as easy in this case to predict what's going to come to the left of any given context because we sprinkled in these mutations, but we still get a pretty good compression ratio. So we compress 64 letters down to 21 runs, compression ratio of a little over 3. Okay. If this is really going to work, then we need it to work for something like human genomes, okay? And in a way, human genomes are almost like a sort of best case scenario because humans are so similar, right? Even unrelated humans are something like 99.6% similar. So if we were to apply the Bros-Wheeler transform to many, many human genomes, would we be able to get these good sorts of compression ratios? Let's take a look. So I'm showing you latest greatest data here because this is up to and including the latest release of the Human Pangenome Reference Consortium, which was earlier this year. So the Human Pangenome Reference Consortium is using long reads and careful, good, expensive sequencing to produce lots of well-sequenced human haplotypes. Their most recent release, which was earlier this year, produced 466 human haplotypes. If we think through if we index these 466 haplotypes, or I should say, if we just use the Bros-Wheeler transform on these 466 haplotypes, just imagine we've concatenated them all together, kind of like row your boat 3 times, except it's 466 haplotypes, then the number of runs that we get is about 5.25 billion, all right? Which if you compare it to the number of letters, which is to say the number of nucleotides in all those genomes, means that this is only about 1.58 times as many runs as we would have had if we had indexed 1 genome, right? So we actually only paid for the price of about 60% larger index, we indexed 466 times more genomes, okay? So if we think of each of these reference genomes as another opportunity to match the genetic variants that are in the donor, right? Each new haplotype is another chance that we can have a good template, a good reference for our donor. Then we essentially gave ourselves, 466 swings at the pitch uh, for the cost of not one, sorry, for 1.58 times the cost of one swing at the pitch. Okay, so at least in theory, just looking at what it does compression-wise, the Burroughs-Wheeler transform gives us access to huge numbers, the ability to index huge numbers of human genomes, and we're not even paying that much more than we would have paid to index just one of them, which of course is what we've been doing for all these years. So the cost of admission is not high for getting the Burroughs-Wheeler transform into pan-genomes. Okay, but I don't want to be too self-centered and talk only about humans. There's many wonderful pangenomes coming out, long-read assembled pangenomes for many different species, including yeast maize, Arabidopsis, potato. If you look at their compression ratios, you generally do not find that it's quite as favorable as for human. Humans are a best case scenario for what we're talking about here. But they nonetheless give you compression ratios that you simply can't ignore, right? A compression ratio even of, say 30. Even though we might hope that it would be even larger than 88, just like for the human, it's even larger than 95 here. We might hope that for the potato, the compression ratio would be better than 88. It's not, but it's still a really good number, right? We'd much rather pay this than this than this times, or sorry, than this times 88. So even when we look outside of humans, we see compression ratios we would like to be able to take advantage in order to build pangenome indexes. So is this really possible to do? All I really showed you is that the Burroughs-Wheeler transform, at least in theory, has a way of encoding these pangenome sequences in a way that saves a lot of space. But can we actually index them? And then can we actually use those indexes to do things like the things we do with Bowtie or BLAST or other tools that we rely on for sequencing data analysis? Well, this is the longer story. And I'm going to begin the story by going back to the original sort of paper that proposed using the Burroughs-Wheeler transform as an index. This was an FM index paper, which appeared in Foundations of Computer Science, which is the computer science theory conference in the year 2000. And back then, what it was able to show is that you can answer our two favorite queries, match and locate, with an index that grows linearly with the sequence that you put into the index. All right, so if you gave it one genome, it would be one size. If you gave it 10 genomes, it would be 10 times bigger, right? So linear growth with respect to the input sequences. But not long after that, there was a team that was able to achieve still the same bound for locate queries, but for the match queries, it was able to do it with data structures that fit in big O of R space. Okay, the big O notation, it's not so important what it means. Just think of it as proportional to, right? So the size of the index is proportional to not n, where n is the length of the input, but r, which is the number of runs, right? So this is the post run length compression number, right? This is the good number, the nice small number, right? So this data structure supporting match queries only needed big O of R space, proportional to R space. So great, we were sort of halfway there at this point. We just needed to also be able to do locate queries. We can't do without those, right? The locate queries are important. We need to know where the actual matches are in order for this to be useful in alignment tools. Okay. And how it achieved the big O of R space is, again, not something I have time for today. And again, is something that I would point you to the YouTube channel to check out. But basically, it has to do with clever ways of taking this run length encoded representation, decomposing it into a collection of strings and bit vectors, and then each of those, with some cleverness, can be made to fit in a data structure that grows like R instead of like N. It's not clear from this picture at all because, for example, this bit vector here has length n and this bit vector here has length n, but they're both sparse, and it's the sparsity of the bit vectors that lets you compress it all the way down into R space. Okay, so it only took 5 years to figure out how to do match queries in big O of R space. So hopefully, maybe it'll only take another 5 years to do the locate query, but nope, took longer than that. Took another 13 years. And it was actually only about 7 years ago that we finally had the theory that told us how to do both the match queries and the locate queries in an amount of space that was proportional to R. And it was at this point that the theory finally was opening the door to our ability to really index these pangenomes and benefit from the fact that we get these amazing compression ratios when we use the Bros-Wheeler transform. The additional insights that led to the big O of R on the locate query, again, a bit detailed for our discussion here today. Again, you can check out YouTube. I've got videos on it. This is not my work, but I have videos that explain it. And in my Bros-Wheeler indexing playlist on YouTube. But equipped with this final result, actually the theoreticians that participated in this result, including especially this guy here, Travis Gagey, who's a computer scientist at Dalhousie University in Canada, they were very excited and they said the genomics people, they need to know about this. And Travis started emailing people saying, read this paper. We think you guys can really use this result. And I was one of the people who replied. I may have been the only person who replied. And I started working with this group because I thought, this is almost too good to be true. Is it really possible that we can have indexes just as powerful as the indexes we've been using, like Bowtie and BWA indexes, but they'll work with pangenomes? Well, step one was we have to actually build some of these things. We have to build some indexes over human genome sequences. This was a lot harder than it sounds. So even though we knew what the data structure was that we wanted to build, that doesn't mean you can build it in a reasonable amount of time and space. And it took some creative thinking, especially this prefix-free parsing idea, which came primarily from Christina Boucher, a researcher at University of Florida, and Travis Gagey and others. It took this new algorithm to even get us to the point where we could build pan-genome indexes over human genomes. But once we did, we could see what they could do. And so now I want to show you a series of results that build on this and show you what these R index-based indexes can do today, because they can do quite a lot. And to frame this, I want to talk about sort of three levels of ambition that we might have for these data structures. The first level is substring matching. Okay, so in other words, if I have an index over a pangenome, I would definitely like to be able to ask that index for any possible string I could give it, please tell me whether this string occurs in the pangenome. It would even be great if it could tell me how many times that string occurs in the pangenome. And even where it occurs in the pangenome. That's just substring matching. I give you a string, you tell me where it occurs. That's one level of ambition. The next level might be classification. So maybe I have not just one genome or even one species of genome, but I have multiple choices for species that might be the species of origin for some sequence. I'd like the matching algorithm to be able to tell me which is the best hypothesis, like how should I classify a particular sequence, right? So this is another level of ambition, and it generally is going to require more sophisticated queries than just substring matching queries. A third level of ambition would be just solve the alignment problem, right? Do whatever BLAST and Bowtie and BWA and Minimap and those wonderful tools are doing. Just do it with a pangenome index, right? That's kind of the ultimate level that we might ask for. And since the thing I'm talking about is the R index, and since everybody here knows about Pokemon, I'm just going to call these Rmander, Rmelian, and Rizard. So those are our three goals. We want to see if we can get all the way to Rizard. And I'll just have to break it to you now. We are not yet at Rizard, but I have preliminary things to show you about our ability to reach it perhaps soon. So starting with Rmander, can we do substring matching? Here is a figure. It's maybe not my favorite figure ever because it's pretty busy. So I'm going to sort of gray out a bunch of it. But here's a figure from our paper where we first produced R-index pangenome indexes over human genomes. And what we were able to show is essentially they are eventually much smaller than Bowtie indexes. This we knew we were going to find, right? Like this is a confirmatory result because we knew that eventually R was going to be much less than N, right? We knew the compression ratio was eventually going to be very big. And so that is indeed what we find. And in fact, the green line stops here because we stopped being able to use Bowtie to index larger collections than that because it's just growing linearly. So that actually, and then furthermore, we wanted to investigate speed. This is one of the speed results. This one happens to be concerned with the locate query. And what we found was once we get to a large enough redundant collection of sequences, the locate query actually ends up being faster for the pangenome index than it is for the non-pangenome index. Okay, we might look at this and be like, well, it'd be great if they were crossed even sooner than this, but this was sort of an initial result where we had a relatively theoretical piece of software and we were comparing to a pretty highly tuned piece of software. So we were pleased to see any point where the pangenome index was actually faster than the non-pangenome index. That more or less takes care of Armander. We can do it. We can do substring matching in a pangenome index for sure. That's a I think well well established capability that we have now with our pangenome indexes based on R index. Okay. So on to our Melian, right? On to the ability to do classification. This actually is an area where I think we've had the most interesting results. And this pivots on a paper from 2022 by Max Rossi and Marco Oliva, where they showed how to do a particular kind of query with the R index. And this is an important kind because it helps us get what are called matching statistics and maximal exact matches or MEMS. And so you can think of matching statistics as sort of like at every single point in my query, what's the farthest I can extend it in a certain direction and still have an exact match to the pangenome. And so these triangles are sort of tracing out those matches getting longer and longer until they can't be extended anymore, and then they go back down. And the triangles are overlapping. And the overlapping triangles can also be thought of, by the way, as maximal exact matches or MEMS. And because Moni is capable of performing this query, you can think of it as answering an even more general question than the substring matching example, because whatever substring presence or absence question you have in mind, it's really just a special case of this, right? So if you're interested, for example, in how many tenmers two things have in common, well, you just have to look for all the tenmers that are fully contained within one of these triangles, right? So knowing these matching statistics or knowing these MEMS gives you, in some sense, an index that in turn answers every possible substring query. So it's a pretty powerful query for us to be able to do with respect to pangenomes. Okay, so there's a cascade of work that came after that, too much to talk about, but I did organize it all on this one slide, and I want to highlight that it's all pushing in a certain direction. We're trying to get to the point where our R-index-based methods, our pangenome-based methods, are competitive with alignment-based methods in terms of how well they can do classification, right? Because alignment-based methods, let's say your alignment-based method is like Minimap or Bowtie or BWA, you have no choice but to do Minimap or Bowtie or BWA to many, many, many individual genomes, right? So you can imagine you're either querying over and over again, or you're using an index that's getting larger linearly. Neither of those are very attractive. So if we can actually do better or do as well in terms of classification accuracy, we're also going to be able to beat it on efficiency in all likelihood because we're taking advantage of this compression that they are not. So I'm going to highlight just a few nodes of this diagram. This is the journey I want to talk about today, from Spumoni to Movie to Colby WT, to get a sort of impression of what we can do now. Okay, so let me start by talking about what I'm going to call collinearity statistics, basically any information we can get that has to do with how long are the matches between my query and my pangenome reference. And we already talked about finding substring matches, and we already talked about finding matching statistics. We developed another version of this called pseudo-matching lengths. They're actually not as high fidelity as matching statistics. That we would actually prefer to have matching statistics. But there's some clever ways to make sure you can get PMLs at far less cost than what it costs you to get the matching statistics. So we use them sometimes for our classification problems. And I'm going to be showing diagrams that look like this with all these triangles. Remember, the triangles are the matching statistics peaks. And so larger statistics, meaning longer matches, are taller triangles. Okay. So with Spumoni, the insight that we had is these things are called matching statistics. They're not necessarily anything formal in the sense of what statisticians would call statistics. That sort of comes from the pattern matching literature, this use of the word statistics. But on the other hand, they sort of are statistics in the sense that if a sequence is related to the sequences that are in the database, then the matching statistics should be longer than you would expect by random chance. They should be longer than you would expect if your query was a random sequence, for example. So if you had a notion of how long random or null matches are, then you have a method for doing classification. Are my matching statistics longer than the ones I would expect by random chance, longer than null statistics? And so Spumoni is just built on building a classifier that finds matching statistics. Really, it's finding those pseudo-matching length things I mentioned before. as quickly as possible and then doing a test to see are these significantly longer. And so, for example, if our read and our index are a good match for each other, if my read is truly from a human and my database is also human, then my positive matches will be long. They'll have this long tail relative to my null matches, which will be concentrated around whatever a typical random match length is. On the other hand, if I'm querying with sequences that aren't present in the pangenome, then the null statistics and the test statistics, the positive statistics, will be overlapping and indistinguishable, and you would therefore not classify it as belonging to that group. It just gives a really simple way of turning the immediate output of our pangenome query into a classification decision. And what we found was even this very simple way of doing things was vastly faster and had lower memory footprint compared to an alignment-based method. This is no surprise, and it is not a knock on the alignment-based method. It's just that because we're not doing alignment, right? We're not doing anything like chaining seeds or doing dynamic programming or any of these things that aligners have to do. We're just making this very coarse-grained decision. We can, of course, do it much more efficiently. But the accuracy did, I would say, approach, but came in substantially under the accuracy of the alignment-based classifier. Maybe not too surprising. Again, the alignment-based classifier does way more stuff, right? It's good at finding seed matches, chaining them together, and filling in the space between. But we weren't satisfied. We wanted to see if we could get results that were even better. And it wasn't too hard with at least one change to the index. And the change that we made was, in addition to the index that would answer our matching queries, we also had the index take on some colors or some labels. So you could imagine if we know ahead of time that our sequences have certain chunks of conserved sequence in common, right? One way of thinking of these would be as what we might call multi-mums, so maximal unique matches, but that are present in every sequence. So we could say, well, that's like a landmark, like that's every sequence has the same landmark. So maybe I'll color that landmark a certain color. So maybe if I color this one red, this one blue, this one orange, this one purple, and if I can also detect when my matching statistics are falling within one of those landmarks, then I get not a fine-grained statistic, but more of a coarse-grained statistic, because now I can start to see if many of my triangles all seem to have a consistent color, consistent with the fact that they're coming from a collinear stretch of one of the sequences in the pangenome. So looking up here, if these are the triangles that I find in a particular query, I might say, aha, look, these aren't perfectly red, but they're mostly red. These aren't perfectly blue, but they're mostly blue. That probably came from a piece of the pangenome that spanned those two conserved regions. And the fact of the coherence of these colors is extra information that this looks to be the true point of origin for that read. This one would be unconvincing, right? This is kind of a mix of different colors and the statistics are a bit shorter, so maybe we'd be not convinced in that case. So although we're not formally doing anything like chaining, right, which is what Minimap is actually doing, we're doing this kind of coarse-grained, roughly something like chaining, right? Because we're noticing when the matches seem to be collinear with respect to the reference. And it's at that point that we can finally, again, I'm not going to claim that we've reached it because we're a little bit better on specificity, but we are a little bit worse on sensitivity. But we're now close to indistinguishable with what an alignment-based method does. And it is definitely far, far, far faster and uses less memory. Okay, so this is a good example of what we couldn't do this before. We didn't have pangenome indexes until a few years ago. We could not do this before. Now we can do it in a way that's actually very favorably competitive with an alignment-based method. So good progress. Our Melian is well on its way to being a powerful Pokemon. One other thing I'll mention is I highlighted another tool called Movie. I'll talk about this very briefly. Again, I'm going to point you to YouTube because I have a whole series of videos on this topic known as the move structure. But another wonderful theoretical advance that's only about 4 years old is a proposal for how to modify the R index in a way that makes it far, far faster. And I was talking about this with the students this morning. It actually wasn't invented for that purpose. It was invented to slightly improve one of the theoretical mathematical bounds. But in the process of doing that, they made something that's a much better fit for the typical modern cache hierarchy on a computer. And so it's actually vastly faster. So a huge part of what we're able to do today in terms of speed is because we're using this move structure adaptation of the R index, and it incurs only a tiny number, something like the minimal possible number of cache misses per inner loop iteration. those cache misses can be further hidden using latency hiding approaches like memory prefetching. So we're sort of finally at the point where we're doing just about as fast as we could possibly imagine doing in our inner loop while still having a very powerful pan-genome index. Movie caught the attention of at least one other lab. So this is Rob Knight's lab at UCSD. They do a lot of work with human microbiome, and they're, of course, therefore very concerned with filtering out host sequence, right? Whenever you deal with human microbiome, you want to filter out the human reads for privacy reasons and then also for getting correct answers downstream. They saw the movie paper and they said, that's what we need, right? Because we are not convinced that the screening that we're doing that's with respect to, say, the CHM13 genome or any other small collection of reference genomes is going to be good enough to really remove all the host sequence. So they said, can we please use movie? And they integrated it in a few different ways into their pipeline. And they decided that at least one of the pipelines based on movie, can't quite remember which one they said they preferred, but was what they were going to use from then on. And so they have a paper saying this is our preferred pipeline. You should use this thing called movie, which again is a pan-genome index that uses the move structure and the classification ideas that I was just talking about. So it's found its way into practice for real practitioners already. Okay, that was the version of what we can do now. And you'll recall we got partway through the story, right? Armander and Armelian are up and running. We can do exact matching and classification with pan-genome indexes. Let me now say a few things about why this excites me so much. So first of all, these are not K-mer-based methods. I just want to make that perfectly clear, right? So these are full text indexes. Full text indexes don't require that you choose a parameter K and shred everything into K-mers first, which is really good news, right? We don't want to do that. We paid a lot of money for these wonderful genome assemblies. Why would we shred them up into K-mers first, right? That's not a good idea. We should be able to use all the contiguity information that we paid top dollar for when we assembled these genomes. We have excellent speed, probably pretty hard to do better on speed based on just how tuned the inner loop is. And we can always wish for better, but it's hard to imagine a better use of, for example, the memory hierarchy than what we can already achieve with movie. And it's vastly smaller than what a bow tie index would have been because we're taking advantage of the compression. However, we are not in the promised land yet with everything that we can do with pan-genome indexes. For one thing, alignment level classification, even with all our improvements, was still slightly more accurate, right? So in other words, there's probably still a few more things we can do, things that approach the full feature set of an alignment-based tool to get all the way there and to do it with higher speed and lower memory footprint. Okay? So there's more that could be done, but we are, I hope you would agree, very close. So I think the closeness is already very appealing. We do not yet scale well for read alignment, and I'm going to talk about this again in a moment. This has to do with how seed and extend works, and the fact that seed and extend in pangenomes can be a very repetitive and frustrating thing to do. And actually, it doesn't scale very well with the size of the pangenome unless you do something clever. And so we're still in this fix right now. And it's not as small as K-mer-based indexes. So K-mer-based indexes are still smaller than full text indexes. But this is probably unavoidable in the sense that K-mer indexes are happy to discard a lot of information, right? K-mer indexes discard any notion of contiguity beyond that length K. Full text indexes keep all the contiguity, right? You don't have to pick a K. You might be interested in K equals 7 today and K equals 1,000,000 tomorrow, and we can satisfy all your queries with a full text index. But for a K-mer index, you have to pick one and you're stuck with it. So this is perhaps not surprising. Okay, so we do not yet scale ideally for read alignment. This is avoidable, I think. We're still not as small as K-mer indexes, which is perhaps unavoidable, again, because it's not reasonable to think that a single K-mer value, that a full text index, which is capable of handling any length K-mer, is going to approach the size of a K-mer index that specializes in a single length. However, I want to talk a little bit now and conclude with what I think they can do in the future, and it's going to include some preliminary results on the R-is-ard front. Okay, this slide is too busy. This slide is also too busy. So I made a cartoon of this slide, and I'm just going to talk about this slide right now. So in our experiments so far, so we have an implementation of a full aligner. So it's an aligner that behaves just like BWA except it's using a pangenome index where the pangenome is a collection of linear sequences. And this is primarily the work of Rahul Varki and Eddie Farrow and Max Rossi, and it's primarily from Christina Boucher's lab, again, at the University of Florida. And this is available today. You can use it. It's called Moni-Align. Okay, so Moni-Align exists and you can use it today. But it has a slight problem, right? Which is that it doesn't actually scale with size of the pangenome very well. And you can see this most apparently if you compare it to graph aligners. So graph aligners, which also use a pangenome index, are not scaling like this. They're scaling more steadily, right? You can grow the size of the pangenome and it doesn't make the tool much slower. But for Moni-Align, it does. And this is because graphs collapse redundancies in a way that we are not collapsing redundancies when we include all the linear sequences in our pangenome index. It's a trade. When you do collapse the redundancies, it's usually because you started from a multiple alignment. In order to start from a multiple alignment, you usually needed to discard some things, right? So when people put human genomes into multiple aligners, it doesn't work. You have to take away some of the more tricky structural variation and repetitive sequences, then they'll multiply align in a reasonable way. So you are losing something with the graph aligners, but they have done a kind of collapsing that's extremely favorable to performance. So the collapsing is a good idea if you want to do alignment. And we are not doing that yet. So we are on this line that has not so good scaling. However, I'm sorry, I put these in a weird order. However, I do think that there is a technical solution to this problem, which is something called BWT tunneling. So Burroughs-Wheeler transform is capable of doing similar kinds of collapsing as you would be doing in a multiple alignment, but without using a multiple alignment and without losing any information, right? So this is a transformation that can be done directly on the strings once they've been concatenated. And in a series of steps, it can produce something like a compressed index which has collapsed all of the perfectly conserved regions across the genomes in the same way that will reduce the number of locate queries that's making us slow. So that's one thing that I would say. We're not yet at RSR, but we think we can see the technical path to getting at a fully parallelizable, a fully equivalent to the graph aligner performance, a pangenome aligner, but that does not use a graph and does not use multiple alignment. Another thing I'll say is this, is that pangenomes aren't always the right choice, even just if your goal is to avoid reference bias. So let me point out that if your goal is to avoid reference bias, and let's say you have whole genome DNA sequencing data, a better choice is to make a personalized reference. Right? So the reason I say this is because pangenomes, they're inclusive. Inclusive is good because some of the alleles that are in that inclusive set will match the alleles in the individual. However, a bunch of them won't, right? So the pangenome will have tons and tons and tons of rare variants that are not present in your donor. Those can only make read alignment worse than if it had only the variants that are present in your donor, right? If you want to, ideally, you want your reference to have exactly the variants that are present in your donor. And wonderfully today, we have imputation panels, excellent imputation panels, including imputation panels that include lots of structural variation and which are appropriate for building personalized references. So another approach would be you give me a whole genome sequencing data set, I take it, or maybe even I just take some of it, because actually 1x coverage is plenty for imputation. I align those reads to a linear reference genome. I then use an imputation tool to impute a personalized diploid reference for that same individual. Now I align all the reads to the personalized reference. That yields variant calls that are better than what you would have got with a pangenome. So this stands to reason, and it's another option that I think we can be confident reaching for in the future if we want to avoid reference bias. And the other thing I want to point out about this pipeline is if you open up these tools, the tools that do imputation, the tools that do alignment, and look at what's inside, they all have the Burroughs-Wheeler transforms because these days genotype imputation is done with something called the positional BWT, a variant on the Burroughs-Wheeler transform. And both graph and linear pangenome alignment-based tools are also all based in some way, shape, or form on the Burroughs-Wheeler transform. Okay, I covered this. So I think I can actually wrap up, hopefully without going over. My vision is that in a future where we're getting at a regular pace, new, long read, high quality reference genome assemblies, we want it to be the case that more is better. If you give me more assemblies, I can give you a better answer. What we have some obstacles currently to this, which is that, for example, we aren't necessarily using them because we aren't necessarily using pangenome references. So using pangenome references is a huge step in the direction of more is better. Also, we would like our indexes to index just the allelic combinations that are really present. And this is a problem with some graph genome representations. They might allow spurious recombination between variants that are in the graph that didn't actually appear in any of the individuals that were used to build the graph. This creates a kind of blow up in the space of possible alignments and again leads to ambiguity and more incorrect alignments. So I think sticking to the actual string sequences of each of the haplotypes is a better bet when it comes to achieving more is better. That's another reason why I prefer the linear idea of the pangenome. And the last thing we need to do is avoid lossiness. When we've gone through the pain and trouble of assembling high quality long read end-to-end T2T assemblies, the last thing we want to do is shred them into tiny little bits by K-merizing them or do whatever it is we need to do to torture them so that they'll fit in a multiple alignment because they don't want to fit in a multiple alignment. And so approaches, again, that leave the strings as strings, I think, are going to be ideal for achieving this more is better criterion. I've said it many times. This is just on one slide, some different links for talks that you can go to that cover topics that I glossed over today. And then finally, I want to thank all my collaborators, but especially Christina Boucher and Travis Gagey, who were very instrumental in introducing me to the latest advances in computer science that have enabled pan-genome indexing. And thank you for listening.
Questions for, yeah, oh, fascinating work like always been. I remember back when we interacted a little bit more, we also talked a lot about like which kind of variants we should integrate in the pan-genome and which kind of sequences don't make so much sense. And so I wonder about that part, of course, versus the BWA tunneling is quite kind of interesting as well. But like, would that part kind of be destroyed if you just increase the number of sequences that we do? I mean, like, so what I'm saying and what I'm trying to formulate is like, you know, a large proportion of the genome, we can debate how much, doesn't really need a pan-genome maybe. It's very similar, right? But like some parts needs it. And like one downside of the pan-genome to some extent is like, if I'm getting all the humans of the human population in there, you're getting like huge branches and huge problems. Yeah. So like I was just wondering if you can elaborate on that a little bit more.
Yeah, I mean, there is always this tension with pangenomes, which is in order for, maybe another way to say, in order for more to be better, it has to be the case that they truly live in common coordinates together. To the degree they don't truly live in common coordinates together, which is the case because of, for example, hypervariable regions and other things that might happen, then I feel like the policy is do no harm, right? Like at that point, right? Like just keep it the way it is. Don't force it into common coordinates if it doesn't belong there. And also don't get rid of it, right? And that's what I think a lot of the graph-based approaches, they're driven to make the graph as concise and as bottlenecked as possible. And that's what sort of drives them ultimately to get rid of stuff. But the R index with tunneling is, I hope, the best of all possible worlds because the sequences are there. To the degree they have common coordinates, we can exploit that purely for efficiency, right? We're not exploiting it because we're telling the world these things are conserved, right? Like it's just purely an internal matter inside the index that redundancy means that we don't have to do lots of locate queries. We only have to do one locate query. And so we're not passing any biological judgment on like what belongs in a conserved category or anything like that. And we're certainly not throwing anything out. So I hope that to the degree that more is better, we can achieve it. And because again, we're avoiding lossiness and things like this. But there is also a degree to which more is not better because common coordinates are fiction, right? You know, common coordinates are only partly true. And that's, I think, is a more notional thing of that we have to ask ourselves, especially since we all think of ourselves as moving into a pangenome era. Well, what about the parts that aren't really in any common coordinates with each other? Or where it would be a mistake to think of them that way. I do think there's larger questions about that. But so I would say at least we're doing no harm, right? In the sense that we're not forcing them. We're not excluding them, but we're also not forcing them to be something they're not. Yeah. Yeah. Yeah.
Theory for +1 problem for our indices, because I mean, ideally we would want to build the pan-genome index, and
Yeah.
when the new batch of genomes comes in,
Yeah.
we probably don't want to rebuild it.
Yeah, increasingly there is. So I would say there was definitely a deficiency at first in the literature where you basically had to start from scratch if you wanted to add one. There are multiple new efforts. So one of them is very recent BWT merging ideas. You can build BWTs for partitions of the genomes, and then there's ideas for how to merge them. There's even a prefix-free parsing compatible way to do that. But the ideas that seem simplest and to work well enough are like the ones that are used in rope BWT3 and similar tools. There was another one too from UCSC, I forget what it was called. But they just basically do incremental building where the run length compressed BWT is held in a tree structure, so something like a B plus tree. And those are very slow, but they are certainly capable. If you want to solve the N+1 problem, they're ready to do that with minimal pain for the N+1th genome, even if the pain was high for the original N genomes. So increasingly, there are a stable of tools that can do that.
No, but for someone who's not familiar with pan-genomes, could you describe what metadata, if there is metadata, is associated with each genome?
Yeah.
In terms of like disease association and whatnot?
Oh, I think you'd find it very disappointing because I think the sad truth is that there are of course nowhere near as annotated as our favorite long-standing reference genomes, and that the task of moving annotations between genomes in a pangenome also is not in a superb state right now. There are tools that do it, but then upon inspection, they kind of fail at everything that's hard. So having useful annotations for all the genomes in a pangenome, I think, is another major dream and goal that I would argue we're currently very far from. It's not something my lab works on, but I hear it complained about.
Why I asked that question because I think, at least I make the assumption that GRCH38 is the normal human. And I make the assumption that T to T is the normal human, HD0. But then when you have 466 genomes,
Right.
there's going to be individuals in there with diseases.
Yeah. In fact, T to T shouldn't be considered normal either, right? Because it's a mole. It's a molar pregnancy, right? It's not even. So yeah, absolutely. There, I think, is metadata, not annotation necessarily, but metadata was your point. Now I see better. I do think that is maintained by groups like HPRC. I don't actually know that for sure. Actually, no, I think what they do is they sequence cell lines.
They're pretty bad in metadata.
Yeah,
that's one of the criticisms of HPRC, that they don't keep medical records.
Okay, so
yeah. Yeah, so it's
almost got an R1 funded because of that.
It's I guess no better than, say, if you wanted to know disease status of 1,000 genomes. Yeah.
No, because
Yeah.
like, it's all about
Right.
genome cell line.
The goal is to be able to identify variants of significance.
Yes. Yes. Yeah.
And so one possible outcome is losing the forest from the tree,
Yeah.
by throwing too much in there without
Yeah.
knowing what is in it.
Right. Yeah. Right. It's true that when these projects toss in another tranche of genomes, it is, our hope is that what we're gaining, that's good, is that if there is a penalty that we would otherwise have incurred, but that penalty was associated with a variant that actually was known to exist, we would like to take that penalty away because we think that's the reference bias causing penalty. But like you say, if it's just stuffed full of tons of individuals, then the vast majority of those variants are going to be rare ones, right? Which means that they're confusing you a little bit, right? They're making the whole representation more ambiguous, which is again why I think personalization might be the true way to go if it's possible, right? If you have the data, you'd prefer to have just that individual's alleles. And the good news is we can do that too. But you're right, to the degree that the pangenome is going to be useful, it doesn't seem like we should try to get rid of rare alleles, for example, or that we should try to get rid of disease alleles, for example, because we do care about whether they're there and we do want to not be biased against them. But you're right that information not being there creates question marks. When results do come up, how should I interpret that? Yeah.
I have a question. I was just wondering, is it possible or how to align or hurry a transcript sequence? For example,
Yeah.
long read RNA-seq reads against the R index-based pan-genome.
Yeah, that's a great question. So there is, I think, really good work from the VG team. So the VG team has a graph-based pangenome aligner, and they have done transcriptome alignment to their graph pangenome. And so that's probably the best place to look for good answers to that. There's sort of no reason why you couldn't do that with linear reference genomes as well. There's just so many tricks to spliced alignment, though. I think the trick there is if the whole point is to take away penalties from variants that you know might be there, then when you move to spliced alignment, the question is, are you trying to take away the penalties for the splice junctions that you know might be there? But that's very hard because the space of possible splice junctions is going to be, quite large, even compared to the space of possible variants. So I think what everybody would probably do as a first pass, and what I believe the VG team did was, no, we're not going to try to put splice variants in the graph. We're just going to try to put genetic variants in the graph and then still hope that we can find those splice junctions with tricks like looking for anchors on either side and filling in the middle. But it's definitely something people want to do. I would also love to be able to do the personalization approach for RNA-seq. I think that's entirely possible, even though we won't be able to personalize the entire reference. We'll only be able to personalize like islands around the expressed genes. And even that will be a little tricky because there will be allele-specific expression, which means the hets won't quite look like hets. But I do think that should be possible too. But I don't know of anybody, well, I shouldn't say, there probably are papers that are trying to not only solve the genetic variation problem, but also the spliced alignment problem using graphs. I'm just not quite as familiar with that.
One last question, then all the way in the back.
It's similar to the previous question. For clinical genetics, what do you see the value add to actually improving the reference genome based on the pan-genome?
Yeah.
So like, do you think it's like going to be decreased false positives? What exactly are we going to see?
Yeah, I think that whatever your clinical goal is, so like, for example, my PhD student who did the work on personalization did an internship at Bristol Myers Squibb. And what they wanted to look at was a bias they were noticing that when they did tumor mutational burden and microsatellite instability on their patients, they saw different answers for different genetic, for people who came in who had different ethnicities. That was reference bias, right? That's a reference bias problem. So like if this won't happen, but if FDA tomorrow said we're tired of approving these diagnostics that don't take, that work differently depending on what the ethnicity of the person is, you have to show us that you can overcome the reference bias problem, then people wouldn't have a choice. They'd suddenly have to use approaches like this, right? So I think ultimately that's the goal. We should be able to do, first of all, equally well everywhere in the genome, even hypervariable regions. That's one aspiration, right? So if the disease relevant variants you're looking for in hypervariable regions, this of course is something you want to see happen. But then also there should be no difference in accuracy if you're one ethnicity versus another. And the assay that is determining your course of treatment is a sequencing-based assay.
Let's take a speaker.